Welcome, everyone, today to our seminar, our NOAA Omics seminar series on creatures of darkness, investigating life and light in the Twilight Zone by our speaker today, Heather Bracken-Grissom, who's the Florida International University, a professor and assistant director of Coastlines and Oceans Institute of Environment. We are very excited to have her here today. Before we get into that, though, I just want to highlight NOAA Omics itself. And I am Katie Poser. I'm standing in for Nicole Miller, who is the NOAA Omics coordinator. But I want to thank everyone for joining. Today's presentation, of course, is brought to you by NOAA Omics and the NOAA Library, where I'm in. Omics is a suite of tools. Let me get on the right slide. NOAA Omics. Omics is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series as an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series, as I said earlier, are available on YouTube and posted up on the Omics page as well as the NOAA NOAA Library YouTube. And as participants, you are muted. Feel free to type those questions into the Q&A panel, as I mentioned, and we'll read all those questions at the end. So, Heather, I'm going to turn it over to you, and you can take it away.
Awesome. Well, thank you, everyone. Just to say hello, here's my face, but then I am going to turn my face off and let you see all of the slides because there are some pretty pictures on there. So let me stop my camera and go here. All right. So hopefully everyone can see everything okay. If not, please just shout out because I can't see anything anymore but my slides. But anyways, I just want to thank the NOAA Omics group for having me today. I'm really excited to talk to you a little bit about what my lab has been doing in recent years, introducing you to some of these wondrous creatures that live in the Twilight Zone and talking about their amazing adaptations. So, as was mentioned, I run the Chrystomics lab at Florida International University, and we take this integrated approach. We combine genomics and taxonomy to study all aspects of crustacean evolution. So, at the heart of everything we do is basically defining, documenting, and characterizing biodiversity. I'm going to talk to you a little bit of how we use genetics to characterize biodiversity in the Twilight Zone. And this really lays the foundation for almost everything we do. We're also extremely interested in how these units of biodiversity are related to one another. So we ask questions like how have they evolved across space and time? We use phylogenetics. We're interested about origins and everything are related to their evolutionary history. The third major area and interest in my lab is adaptation. And today I'm going to be talking to you about bioluminescence, which is one of the adaptations that these organisms have evolved to survive in deep, dark habitats. We put this together, and all of these things allow us to say something about patterns and processes that are occurring in the deep sea. And of course, all this research does not happen in a vacuum. I need to introduce my amazing lab. Currently, I have 3 postdocs in the Chrystomics lab, 6 PhD students, a lab manager, and just a whole suite of undergraduates that work with us. And a lot of the research that you're going to see today are this kind of collaborative effort by all of these people. And then also just a shout out to previous Chrystomic members that have gone through and where they have landed today. So as you probably inferred, crustaceans are our model organisms. And this is an extremely species-rich group. Crustaceans, they are non-monophyletic. Of course, with the insects, they They make a monophyletic group called the pancrustacea. But if we think about crustaceans and how they've historically been thought of, there's about 70,000 species. They are economically important, not just on dinner plates. A lot of people love to eat these critters, but they are a really important component of social and cultural livelihood. So I lived in Louisiana. This is where I did my PhD, and I can tell you that crawfish and crabs are kind of central within like celebrating festivals and all of these different events. They're morphologically unique, which is kind of demonstrated by this slide. You can see all of the fantastic shapes and morphologies these animals exhibit. They're ecologically diverse. You can find them from the poles to, all the way down from the Arctic to Antarctica, freshwater, marine, tropical environments. And what makes me really interested is that they are an extremely old group, almost 500 million years of evolution, and they have adapted to almost every condition and habitat imaginable. So they're extremely interesting to study for evolutionary biology. So we have a lot of projects that occur in shallow waters in my lab, but the majority these days are happening in this deep sea twilight zone. And I'm sure most of you are familiar with where this is, but for the purposes of this talk, I want to define the twilight zone. It's an area of the ocean which occurs from about 200 meters to 1,000. And as the name implies, this is where sunlight can still penetrate, and you have this very dim light that exists. Twilight zone is also referred to as the mesopelagic, and it contains a remarkable array of diversity. Some of those animals are here. You can see that we have fish, we have jelly, we have gelatinous organisms like ctenophores and cnidarians. We have a whole suite of fish. We study the crustaceans, but then you also have these deep diving mammals that dive into the twilight zone to feed on all of the diversity that exists there. In this area, it's also where the highest density of bioluminescent organisms reside, and we're going to talk about that in just a little bit later. So this slide kind of demonstrates the wonders of what we get to encounter in the twilight zone. And I just want to take a moment to acknowledge the photographer, Dr. Dante Fenolio. He is someone that comes out on all of our research cruises, and he basically brings these animals to life in these awesome photos. So every photo that you're going to see in the talk today is something that we've collected as part of our expeditions. And again, I don't think there were any cephalopods on that last slide, so just giving some love to the squid and the beautiful octopus that occur in the mesopelagic. Now, these deep sea organisms are really critical to the health of our planet. They participate in the largest migration on the planet in terms of biomass. And this is called diel vertical migration. And I'm sure some of you have heard about this enormous migration. But basically, what this describes is a predator avoidance behavior. So, what happens every single night is almost the entire deep sea community migrates into shallow waters to feed and mate. And as the sun rises, those animals, and as those waters become sunlit, those animals migrate back down into the deep, dark waters. And you imagine that this migration happens because in shallow waters, there's more food. They kind of form these aggregations where they mate. But as soon as those waters become sunlit, the animals are threatened by predation and they need to migrate back down. And this happens every single day over 24-hour period. So it's a huge amount of biomass that's moving up and down in the water column. These regulate our climate through the biological carbon pump. They bring this carbon, they sequester carbon at the surface, they bring it back to the depths of our ocean. About 90% of organic carbon from the surface is respired back into carbon CO2. This, really fuels this kind of connection from shallow to the deep sea. So they move nutrients, they move all kinds of things, and they are absolutely essential to the functioning of our oceanic systems. So, the first part of my talk, I'm going to describe how we've been documenting biodiversity in the Twilight Zone and how we've been using genetics and some omic techniques to document the biodiversity. Part two is going to be the adaptation of that biodiversity, and specifically, we're going to be talking about bioluminescence. So I've been really fortunate to be part of an amazing consortium called the Deep End Restore project, and this is a 47 member, 11 institutional research project program aimed to examine the trends and drivers in the Gulf of Mexico. Specifically, we started back in 2010 after the Deepwater Horizon oil spill, and we were charged with the goal to figure out how the oil spill affected the organisms that lived in the mesopelagic. So, since about 2010, our group has spent over 400 days at sea. It's one of the longest time series collections of mesopelagic fauna in oceanographic history. I joined the team in 2015. That's why I have that little yellow dot there. And I was really kind of in charge of documenting the biodiversity of the crustaceans and using genetic methods to document this biodiversity. So we're attempting to narrow this huge gaping black hole of knowledge that exists for mesopelagic animals, and this is what we're going to share with you today. So I like to argue that we really, at least for the Twilight Zone and some of these other habitats, we still don't know what we don't know. And this is especially true in terms of biodiversity or what animals exist in certain habitats. And probably one of the best examples of this is this amazing expedition that I was able to participate in. This was in 2019, and it was called Journey into Midnight. And our team set off into the Gulf of Mexico to document new sources of bioluminescence in the mesopelagic and the midnight zone. And on this trip, we were able to capture something truly phenomenal, and I'm going to show it to you today. So, on that trip, we were able to actually film the giant squid in its natural habitat. And it was only the second time it had ever been caught on film, ever, and the first in US waters. So, why I say that this was kind of a remarkable find was that this was only about 100 miles southwest of New Orleans, and it was in like sighting distance of some of the largest floating oil drilling platforms in the Gulf. So, a lot of people, we still have incredible things out there that we don't know exist, and there's still so much more to explore. So, I would also argue the second reason we need to narrow this huge knowledge gap is that we're losing biodiversity at alarming rates. So, what you're looking at right here is a time series of mesopelagic abundance in the Gulf from 2011 through April 2021. So, this includes a 10-year time series. And what we found is that animal abundances have plummeted. And this is for every single major group, every depth bin, day and night. And this is across these three different groups. So we found that fish have declined by 66%, shrimp by 57%, and cephalopods by 61%. So this paper is submitted, it's in review right now by PNAS, but hopefully that's going to be coming out soon to show this kind of urgency of why we need to continue to document and protect biodiversity. We're also using genomic methods to look at these declines that we found in abundances within the Gulf. So, my postdoc, Dr. Pedro Perez, has found using genomic diversity methods like and effective population size, and he's also used ecological niche modeling, that these crashes in abundance are not only in abundance, but we're finding these crashes in genomic diversity and effective population sizes. So, some of these animals, we did this experiment over 3 important fish and 2 crustaceans. And this is the longest time series of genomic deep sea diversity, I believe, that has ever been done. And we're seeing some pretty alarming crashes in a few species of fish that are really important in the ecosystem. Some things aren't changing, which definitely means that these are species kind of dependent. So we have to look at each of these species one by one to really figure out what's going on. But we are seeing some of this diversity mirrored within the genomic diversity also. So hopefully, this really has kind of convinced everyone that these species inventories, figuring out what is there, it's critical for monitoring and documenting spatial and temporal changes. So this is one aspect that we are very passionate about. We've been working at pretty heavily in the lab over the past decade or so. And DNA barcoding, which I'm sure this group is completely familiar with, really enhances these large scale biodiversity initiatives. So, everything that we've collected as part of this Deep End Restore project has been DNA barcoded. We've used very traditional methods, single gene barcoding, but we're also combining that with genome skimming. So now we've really moved from kind of generating these single DNA barcodes, these DNA fingerprints, to using these genome skimming approaches to get a lot larger portion of the genome that we can use in a variety of applications, which we'll talk about in just a second. So this has resulted in some libraries. We're really trying to build these libraries up for the Twilight Zone, for the mesopelagic. One of my PhD students, Dr. Carlos Varela, published the paper a few years ago on crustacean barcodes in the Twilight Zone. So that's been done. And as I mentioned, we're moving from single barcodes to mitogenomes. And I'm sure a lot of you on the call are familiar with why mitogenomes can be, beneficial, and why genome skimming is also kind of a really nice way to document biodiversity. It allows us to really capture those genes that allow us to discriminate species diversity, things like 16S CO1 in crustaceans and then 12S and CO1 in fish. It's extremely easy to recover from museum specimens. I'll talk about that in just a second. It's cost efficient. Each mitogenome, genome skim data that we get is only about $30 a sample. And from this data, you can do so much. You can repurpose the data for all kinds of different applications. So these are just a few of the crustaceans that we are working on. On this slide, you might see things that look familiar, like shrimp, but you also have things like ostracods and euphausids, krill, copepods, and mantis shrimp or stomatopods. So just wanted to show some of the animals that we've included in our barcodes as part of these initiatives. So, in a similar fashion, we're really using these DNA barcodes to build something that I'm calling anchor trees. And these anchor trees are allowing us to reveal all kinds of new species discoveries. It not only allows us to kind of identify these unknown species that sometimes come up in our trawls and we're not able to tell what they are. Maybe something is damaged, or maybe something is just we've never seen before. So these anchor trees are not used for evolutionary relatedness. These are single gene trees that we have built. What you're looking at on the left is basically a 16S single gene phylogeny of about 100 species of midwater shrimp that we've put together. Every single one of those shrimp are vouchered. So there's a museum specimen voucher that's associated with that sequence, and they've been identified and verified. So that anchor tree is really kind of captures the amount of diversity of midwater shrimp. So a lot of times we find things that are just unknown, like this little critter here. And what these anchor trees do, we sequence the 16S or the mitochondrial genome, or we genome skim it, and we are able to place that unknown kind of in a phylogenetic context of where it should fall in the tree. And this isn't new. This is called phylogenetic informed annotation, and it's been used in a lot of different ways to identify gene sequences or and different things. But we are really finding these anchor trees are helping enormously for biodiversity discovery in my lab. And if you're wondering what that little critter is, because it does look like nothing you've probably ever seen before, this is actually the larval form of this very common benthic deep sea shrimp called Plesiopenaeus armatus. And the cool story here is that that larval form was described nearly 200 years ago, back in the 1800s. And it was only collected a few times in the stomach of a mahi-mahi. And it completely eluded definitive placement. No one knew what it was for nearly 200 years. And in 2012, we found one specimen of this thing in the midwater. And we were able to barcode it and then definitively use these trees to place it, to identify it as this individual. So that's been really fun. And it's been kind of this side project. So oftentimes in midwater trawls, you're going to find these crazy, like bizarre, unidentifiable forms, and you really have no idea what they are. But this is one way we can use anchor trees. And so we've used these anchor trees and we've matched a lot of these babies to their adult counterparts. And it's been kind of a fun side project. What makes this really difficult is a lot of times these crustaceans have a ton of larval stages before they reach adulthood, and they go through these crazy transformations in body shape and body form so that the babies look absolutely nothing like the adults. So this takes a lot of work, not only to match them to the adults molecularly, but you also have to describe these different larval forms and document the diversity. So we were able to do a bit of this a few years ago also and match those, do some adult larval matching using these anchor trees. In other cases, we've used these anchor trees to kind of phylogenetically place, again, just like that baby larvae that you just saw, to place these weird unknowns. This is another strange one called Phycetocaris microthamala. And this, again, had only been collected a handful of times. It was originally, again, identified as a larval form, but it's actually an adult pandalid shrimp. So we were able to place that and also use it to identify cryptic diversity. And just another fun side project I wanted to show, because this has been something that's interesting, is we pull up these we pull up these deep sea shrimp, and they are just jam packed with parasites. So if any of you are interested in omics and parasites, I think there's a wide open field for you all to explore. And this is just a fun example. I was on a cruise, I believe 2 years ago in 2024, and I pulled up this is called Notostomus scabosus. And I was looking at this shrimp under the scope, and this huge isopod starts crawling out from under the carapace. And it turns out that this is a new species of isopod in the family, and it was living inside the shrimp. What you're looking at, I don't know if you can see my pointer, but on the left is the female, on the right is the male. These, we were able to collect both. There was males and females inside of this carapace, and document and work with some colleagues to describe this parasitic isopod. And again, we were able to use barcoding to describe this new species. Again, if you like parasites, I think this is definitely an interesting avenue for exploration. So these are other weird parasites we find on our deep sea surgested shrimp. And I just like showing these pictures because I think they're pretty awesome. We're also moving into the world of fish. I am definitely a crustacean biologist, but we are helping with deep sea fish barcoding, and this has been a really tremendous effort as part of Deep End Restore. Um, Over the past 15 years, we've identified almost 200 new records for the Gulf. And this has increased what we know about fish biodiversity by about 10%. We know the Gulf is already hyper diverse in terms of fish and crustaceans and other organisms, but this just kind of shows how special the Gulf is and why we need to continue to explore it. So, kind of in like, to sum this all up, we just have this amazing collection, and I definitely, open to collaboration, open to different projects. But I wanted to share that we have about 16,000 crustaceans, almost 5,000 fish, and about 250 cephalopods that are in a museum that I run at Florida International University. I call it the Florida International Crustacean and Fish Collection, and these are all collected in ethanol or -80, or we've collected them in liquid nitrogen, but they are all suitable for genetic and genomic purposes. So we're working with this collection to basically document as much biodiversity as we can. This is just a picture of how we digitally characterize that, what that looks like. Every single animal that we collect is curated in a museum in my personal collection at FIU, and it's digitally put into a database called FileMaker Pro. And that database has the whole taxonomic classification from phylum down to species. It has where that animal was collected, all the metadata associated with it, molecular history, location, like what genes we've collected, where that's located, and things. So just really want to reiterate that any sequence data that we get has to be properly matched back to a vouchered curated specimen. And we kind of pride that in my lab. So what we're doing now, we are building eDNA reference libraries. That is an entirely different talk, but definitely want to connect with anyone that's working on similar efforts. But we've been charged and funded by NOAA to build some mesopelagic eDNA reference libraries to improve the reference libraries that we do have for fish, crustaceans, and cephalopods. So that's a huge effort that we have ongoing right now. And last, before I transition into kind of a different theme, I want to plug that we are partnering with the Tree of Life Institute, the Sanger Tree of Life Institute, to kind of launch this new genomic project called the Crustacean Genome Project. This is basically to build these genomic foundations for future crustacean research. There's not many annotated high quality crustacean genomes available. So we are trying to basically rally the community to join us in this effort to kind of launch this new program. So if you're interested, definitely reach out. And we have a Gmail. We've only met a few times. We're just getting this off the ground. Okay, so the second part of this talk is really about looking at bioluminescence, the evolution of bioluminescence in deep sea decapods, specifically shrimp, and thinking about bioluminescence function diversity. And it kind of combines these themes of evolution and adaptation in my lab. So I think when most people hear the word bioluminescence, they hear, I mean, maybe not this group, or maybe not any group that works in the marine environment, but I think most of the general public think this image might come to mind, right? This is the beautiful glow and flash of fireflies during a sunset mating ritual. However, if you are a marine scientist, most of us think about bioluminescence in the form of these beautiful deep sea fish that have remarkable array and diversity of diversity of bioluminescence. You have hatchet fish, you have anglers, you have dragonfish, lanternfish. They have photophores, all kinds of different forms of photophores. They have luminescent tissues. They have lures, some of these elaborate, beautiful lures that are, and escas that glow. You have cephalopods. You've already seen this picture that also are extremely iconic and well known. And if you're more inclined to the animals without backbones, which I definitely am, maybe squid and octopus might come to mind. But whatever image kind of pops into your head when you think about bioluminescence, it's a known fact that this trait is ubiquitous, it's prevalent, and it's an extremely important ecological trait. And this is really evidenced by the ability to produce light has evolved independently about 94 times across the tree of life. This includes taxa like bacteria, which we're all familiar with, and then cnidarians. It's evolved in mollusca, arthropods, chordates, plants, fungi. So it's evolved numerous times across the entire tree of life. But what's really impressive is the prevalence in the marine environment. There's studies that have been done. There's this beautiful study by Martini and Haddock back in 2017 that have quantified the amount of bioluminescence from the surface to the deep. And researchers have indicated that 76% of oceanic marine organisms involved or that live in deep waters off California have the ability to bioluminesce. And this is just an astonishing number to me. So, what you're looking at on the left hand side is this kind of quantification of bioluminescence as you go from the surface down to over 3,500 meters. And there's been recent studies that have traced the evolution of bioluminescence. And this was actually led by the postdoc in my lab, but not done in my lab. This was done in Andrea Quattrini's lab. But that research shows that bioluminescence is believed to have occurred or first evolved around 540 million years ago, most likely in octocorals, so these soft corals. So this dates all the way back to the Cambrian. So really, this pushed previous estimates of the origin of bioluminescence back by almost 300 million years. So prevalent, old. But despite what we do know about the ubiquitous nature of bioluminescence, there's so much more we have left to discover. So, what you're looking at here is bioluminescent ability, which is broken down by taxa. So, that's what the top graph shows. The different colors indicate that gray indicates things that are not bioluminescence. The light blue indicates things that are bioluminescent or likely bioluminescent. And the yellow indicate things that we still just don't know. So, what you can notice almost right away, and I picked my favorite group, the crustaceans, is that there's still regions that are just completely undefined. We just don't know if they have the ability to produce light. But there are some things we do know. And we do know that shrimp, deep sea shrimp, have this remarkable diversity of bioluminescent abilities. And this is, again, something that we definitely work on in my lab. And I wanted to introduce you to the world of shrimp bioluminescence. So, the first mode of bioluminescent in deep sea shrimp is in the form of a secretion. And they used this as a bioluminescent smoke screen to deter predators. So this is made in an organ called the hepatopancreas. It's autogenic, so these shrimp have all of the machinery that they need to produce their own bioluminescence. Bacteria is not needed in this bioluminescent reaction. And they produce this bright bioluminescence in the hepatopancreas. It's equivalent to our liver. And then when they get scared, they emit it through their mouth and through the base of the antenna. And you can imagine, well, I want to show you what this looks like in situ, because was filmed in the lab in kind of this V tank, and that's the actual color. It's that bright. It's one of the brightest bioluminescence in the deep sea. But what does this actually look like? So in 2019, we were able to actually get this on camera, which I think it's really interesting to see what it looks like in situ. And this is what it looks like. And I'm going to show you that just one more time. So we kind of had this bioluminescent lure. This is the same lure that we used to film the giant squid. But we got this, you see this deep sea shrimp swimming, and they use this kind of tail flip response when they get scared. But you can see that it hits the camera, and you can see that smoke screen it emits. So that's what it looks like in situ, which is pretty nice. They also have this second mode of bioluminescence called photophores, and these are light-emitting organs. And these come in a variety of flavors and forms. Typically, these are kind of found, these light organs can be found all along the body of the shrimp. And they are typically dermal, so they're found on the outside, and they can be either lensed or unlensed. The top is an example of some unlensed photophores that are found on the antennal scale of a deep sea surgested shrimp. And on the bottom are this other form, these lensed photophores, and they create this bubble, like this concave bubble around the photophore. We're not really sure why or what the difference, like why there's these different forms, what they're actually using it for. But what we have found, which is kind of interesting, and we're thinking a little bit more about, is these lensed photophores. The animals that have these bubble photophores are typically associated with like this benthopelagic. They are typically benthopelagic species, so associated with slope or the benthos. So we're not sure. Maybe these have an evolutionary advantage in that kind of environment. And the third type of bioluminescent mode is another form of photophore, but these are internal photophores, and they're called organs of Pesta. And these are modifications of that organ I told you about before, that hepatopancreas. So what you're going to see here is I dissected out that light organ, and this is a hepatopancreas. Again, I don't know if you can see my pointer, but it's right there, smack in the middle of the slide. And all that dark purple, black area is the part that that glows. That's the light emitting part of the organ. And I'm going to talk a little bit about the function in just a second. But we've been spending a considerable amount of effort documenting this diversity in my lab and just figuring out like how many species of deep sea shrimp can actually bioluminesce. This resulted in a very nice literature review that was headed up by one of the students, PhD students in my lab, Stormy Collins, who's now a postdoc. And we were able to review all of this kind of the literature, and then we were also able to incorporate all of our observations. And if you want to know more about what species bioluminesce, how many families, what type, you can check out this review that was published in Biological Reviews in 2024. And it was really fun. So this was just one of the kind of pictures that shows all of the different diversity of shrimp and their different modes of bioluminescence, whether it be a secretion or photophores. And we found that over 150 species, we were able to document 150 species of shrimp across 12 families. This increased the number of families from 5 to 12, and we were able to increase the number of species by over 65%. So this just shows how much is still completely undocumented and how much we really have to learn. So this really is, for me, the norm in the deep sea is, the amount of species that can actually bioluminesce has to be, and definitely is, a lot higher than any previous estimate that we think of. We're also tracing the evolution. So now that we know what species of shrimp bioluminesce, we're tracing the evolution of bioluminescence across a brand new shrimp tree of life. So we have about 1,000 different taxa in our new shrimp tree of life, and we're looking how has bioluminescence evolved across this tree. Um, we have about 92, I think more about 95% of families represented, and we have about 28,000 base pairs of data. So we're going to integrate this with fossils and time the origin of bioluminescence. And after we've traced this, some of our preliminary results are showing this single origin of bioluminescence. So most likely within this midwater ancestor, single origin of bioluminescence, but then the different types and the different modes evolved independently or were lost multiple times across this tree. So obviously, we're really interested in documenting the diversity of these luminescent structures and systems, but we're also starting to think about what they're using bioluminescence for. We know that the production of light is used for defensive, so for protection. We also know that it's used for offensive, to maybe attract mates or to lure in prey, and also different mating strategies in the deep sea. So we're starting to think about, well, what is the function of these different modes in our system? And been known for a very long time, actually, that these photophores, those internal and those external photophores, I'm not talking about the secretion now, I'm talking about the light organs. They function in this deep sea game of hide and glow called counterillumination. And counterillumination is basically a form of camouflage in which the animals can precisely match that of downwelling sunlight to hide their silhouettes from predators below. An example of this is this cookie cutter shark, down on the left hand corner of the slide. And you can see that, you can imagine that if you're an animal and you're swimming below that cookie cutter shark and you're looking up, when those ventral photophores are turned on, it camouflages their body against the silhouette of downwelling sunlight. So they use this to camouflage themselves in the deep sea. And a really cool fun fact that I've learned is that counterillumination has only been experimentally shown a handful of times. And one of those times is actually in deep sea shrimp in this species called Eucergestes similis. And this was able to be experimentally induced in the lab. And this resulted in a really beautiful publication in Science in the 1970s, where they were able to put these shrimp in this experimental chamber and force them to counter illuminate. And that turns out to be extremely hard to do. And it's only been shown a handful of times, but the first time it was shown was in these deep sea shrimp. So we're really interested in these photophores in counter illumination. And in recent years, we've been interested in, like, we've asked the question, do these photophores have an expanded role in camouflage or in anything, in how these organisms survive in the deep sea? And we found that these photophores, they not only emit light, but they can actually detect light. So they're not only light emitting, they're light sensing. So this got us really excited. We performed experiments where we exposed light organs to different intensities of light, and we found cellular damage. We've labeled genes in the cell using immunohistochemistry to look for genes that are responsible for light detection. And we've run a lot of RNA-seq and transcriptomic studies to try to identify genes that are involved in phototransduction or in detection of light. So in the photophores, we are finding these genes called visual opsins, and these are a pigment protein. They're known to play a role in light detection in our eyes, but we're finding the same exact genes throughout the photophores also. And now we're really interested in what is what is the function of this adaptation? So we think, so again, we were finding identical opsins in the eye and the photophores. And we think that these are being used to fine tune maybe bioluminescence. So they're almost using their entire body as a light sensor to fine tune how strong of light they emit to precisely match that of downwelling sunlight. We also think it could be a cue for diel vertical migration, or maybe even to help orient themselves in the water column. But it's a really cool adaptation and something we're interested in a bit more. And when you think about sensory systems and structures, you need to think about the sensory organ in different ways, how animals are perceiving a signal. So you think about the signal itself, which is coming out of the light organ, but you also have to think about how that's being received. So we think a lot about visual systems, and the eyes of these photophore bearing shrimp. And we're finding that the visual systems of the shrimp that have light organs are extremely interesting. So what you're looking at here is some cross section of the retina and of these shrimp that either lack photophores. So Acanthophyra purpurea does not have photophores, does not have light organs. And then on the other side, you have Cystilaspis debilis, which is a photophore-bearing shrimp. So these are pictures of cross sections of the retina where we've stained these visual opsins using immunohistochemistry. So if a visual opsin is expressed, it glows a particular color. So we're finding that and so automatically you can see that these photophore-bearing shrimp are expressing more of these visual opsins, and they're expressing different types. So we're starting to ask, well, what's going on? What's going on in these photophore-bearing shrimp? Why are they expressing all of these different visual opsins? And we're finding this in multiple photophore-bearing shrimp. They're not all the same. In Janicella spinicata, it actually expresses this third type also. So there's a lot going on here, and we're starting to dig into it a little bit deeper. So we're starting to come up with this idea that these photophore-bearing shrimp have this expanded opsin suite in their eyes to receive some kind of signal. So we know that the animals that have these photophores, they participate in this diel vertical migration. In some cases, they go from really deep, thousands of meters, they can vertically migrate in a day. And we're finding that these visual opsins are adapted to detect these environmental gradients. So in a study back in 2021, we looked at differential gene expression and how these opsins, these genes were differentially expressed across day and night within eyes. And what we're finding is that we're finding differential gene expression in this one visual opsin. It's called mid-wavelength sensitive 2. And we know that these are sensitive to blue wavelengths. So this provides some evidence that the visual opsin expression is linked to this spectral tuning during diel vertical migration. So could they be using these visual opsins in the eyes to help them kind of detect the light gradient as they perform these tremendous migrations this is one hypothesis that is an interesting to think about. And then the last thing I'm going to talk about before we sum everything up is we've also thought about how are these animals, like, are these visual systems actually adapted to be used in conspecific recognition? So are they using bioluminescence to help with mating or to recognize each other? And how are these visual systems adapted to detect these bioluminescent signals? So just kind of in a very simple question, it's like, how well are these eyes to resolve these point light sources? So this was led by a previous postdoc in my lab, Dr. Schweikert, who's now at UNC Wilmington. And we were able to model this looking at the eyes of hundreds of individuals. So we took a bunch of measurements and we were able to model sighting distance and visual acuity of these shrimp eyes. And what we found was that the shrimp can actually see these point light sources quite far. They can see from anywhere from about 2.4 to 5.6 meters, which is about 63.5 body lengths of these shrimp. But when we looked at the visual acuity, so how well can they actually resolve all of these different photophore patterns, we found that they were actually quite horrible at resolving patterns. So to sum this all up, we were able to figure out that they can absolutely detect bioluminescent signals. However, being able to resolve patterns is likely not possible within these deep sea groups. And then the last thing that's just kind of interesting, and it kind of just, it might be confusing because this is definitely a lot, but we're also finding that these shrimp that have light organs have larger eyes. So this does also suggest a role in bioluminescent signaling. So we know that photophores play a really important role in counterillumination, but we're starting to get more and more evidence that these light organs probably play a role in other important things also, such as conspecific recognition. So just to reflect on some of the things or points from today, I just want to highlight we still do not know what we do not know, and continued foundational studies of biodiversity that couple taxonomy and genetics are absolutely critical. Light organs definitely have an expanded role beyond counterillumination. They probably are being used to sense the environment in some way. It could be a cue for migrations. It could be a cue for conspecific recognition or some kind of signaling. Again, we're not really sure. Last but not least, crustaceans are amazing and gorgeous, and I am here to take any questions you might have. Just a huge thank you to funding sources and all my collaborators part of this project. So with that, thank you.
Thank you so much. Okay, we're going to both come back on camera here.
Here I am coming. Okay.
Wow, thank you so much, Dr. Is it, you have a, is it Dr.
Just call me
Bracken-Grissom,
Dr.
right?
BT. Like, my name is too long. I should have never hyphenated it. Like, this was something I thought was a smart decision. So everyone just calls me Dr. BG. That's great. Or Heather is fine too.
Well, thank you so much for that illuminating presentation and the amazing photography. Like, that was
Right.
very, very cool. And these are species that we do not get to see every day in all their kind of glory, all of their, transparentness. It was very, very cool. So our first question, and we don't have a ton of time, so we'll go through these as they come in, and maybe we won't run out of time, but here's our first question. Given that bioluminescence has arisen so many different times, how different are the biochemical mechanisms? And are they all variations on a basic luciferous
Yeah.
reaction?
Yeah, that's a great question. So they aren't that different. So they are they have this basic luciferin luciferase reaction, 100%. What differs is the substrate or that luciferin. So a lot of the animals that I talked about today have a luciferin called cilantricine, and this is the same luciferin that you find in a variety of different organisms like jellyfish also. So you see this conserved substrate across the tree of life, which is interesting. And the luciferases are also, there's different copies of these. And this is something that people have built gene trees of these luciferases, and they're finding that they're more variable than we've thought about previously. So there's definitely a conserved element to bioluminescence across the entire tree of life, but there is these variations that exist.
Okay, thank you. So take that question down. And my question is like what is next? What's happening in the lab right now that you are excited about?
Yeah, I mean, we're just, well, we are looking at, I guess we're going to, we've moved into hydrothermal vents. So we have an expedition in 1 month where we're going out to the vents in the Caymans and we're looking for bioluminescence at hydrothermal vents. It's never been documented before. So we are doing that. And, excuse me, sorry.
All good.
And we're exploring, we're looking at the visual systems of these vent shrimp. They have these amazing photoreceptors on the back of their carapace, which are thought to maybe be able to detect black body radiation. So we're looking at how these sensory systems on these shrimp, like how they're using them.
Very, very cool.
Mm.
So we've had you talking for too long.
Yeah,
Um, we,
with my throat.
I think we have I think this is a follow-up from the first question about the bioluminescence. Wouldn't the conserved element indicate a single origin?
Yeah, so yes, yes, but it's been modified several times. So so yes, but also, um, I mean, if you look across the tree of life, you could interpret this as several losses, or that it's evolved several times. Because if you look at across the breadth of, of the phylogeny indicates that it's evolved 94 independent times. So there's definitely some convergence at the molecular level, but the phylogeny does not indicate a single origin unless it's been lost more, more times.
Thank you for answering that follow-up.
Mhm.
We are at 3 o'clock. If someone did have another question, I am sorry. You can always email either the library and we'll pass you on, or I do believe if you captured contact information from the slides, follow up with Dr. GB, a BG here. So thank you so much, Heather. This was amazing. Reminder, everyone, we recorded it, so it'll be up on the YouTube channel for the library, up on the omics as well. And just highlighting that the fact that we will have another omics seminar in July on the 15th, talking about the role of structural variants in defining population structure and repeated local adaptation into cupid fishes. Cupid. I am not the scientist here, so sorry, everyone. Take care and have a wonderful rest of your day.
Thank
Thank you
you.
again.
Bye.